Hello and welcome back to Excelsior Academy. So again, I have come here with the another data interpretation question, which is going to help you in your research analysis and developing the skills. So let's do the data interpretation. So this is based on the regulation of the nuclear lamina by phosphorylation. So before I begin, I would like to just revise certain topics which is related to the nuclear lamina. So if you remember lamina, they are the lamin proteins, which comes under the category of intermediate filaments. Three types are there: A, B, and C. Now, when phosphorylation of lamins happens by the CDK1 activity, the nuclear envelope starts. The destruction starts, that the disappearance of nuclear envelope starts. So, keeping these concepts in the mind, let's do what is given in the experiment and what kind of questions they have asked. Okay, so the experiment says the nuclei isolated from embryonic chicken cells were incubated for the time period indicated in the figure. Okay, so that is 0 hour, 1.5, 1.5 hours, 1.5 hours. Okay, so indicated in the figure with highly purified CDC2 kinase or otherwise CDK1. So 1, 2, 3, and 4. So in these all, you have got your CDK1 activity in buffer without kinase as the control, or with the protein kinase C, that is nothing related to the protein kinase. Okay, so PKC over here. Now incubations were carried out in the presence of ATP. Afterwards, Triton X100, which is actually a mild detergent, was added to the nuclei, and nuclei were fixed and stained by immunofluorescence microscopy using anti-lamin B antibodies. Of course, if you're studying the lamin proteins, then you need anti-lamin antibodies. Now the phase contrast micros corresponding to A, C, E, and G are shown in B, D, F, and H respectively. Okay, so the scale bar is given around 10 micrometer also. Now this figure is there. Okay, now let's do what are the questions they are asking. Okay, so what was the role of ATP in the incubation mixture? Now this is very straightforward question. Why? Because here you're talking about CDKs. Right. Now, what is CDK? It is cyclin dependent kinases. And I have mentioned during the cell cycle lecture series also that kinases means they are going to need ATP. Now, why? Because these kinases are going to transfer the gamma phosphate group to the serine or threonine group of the or amino acid of the substrate. Now, why serine and threonine? In the earlier lecture also I have mentioned because they have got the OH group. So to this OH group of their R gene, the gamma phosphate is going to be added. Now why gamma? So if you know that you have got the adenine and it is triphosphate, right? So this is alpha, beta, and gamma. So it is called gamma phosphate. So this phosphate is being transferred to the OH group of the serine or threonine amino acids of the substrate. And that's why you need the ATP in the incubation mixture, because you are dealing with the CDK. And to carry out the activity of CDK, you need ATP as your incubation mixture. Okay. So these are the because these protein kinases will catalyze the transfer of the gamma phosphate group from the ATP to the hydroxyl group of the serinothreonine amino acid chains of the substrate proteins. So that's why you need ATP in the incubation mixture. Okay, now I'm going to the next question. What is the purpose of the Triton X100 treatment? So we know that it is mild detergent. So this mild detergent is going to make the membrane, the nuclear membrane, since we are talking about the nuclei over here, it is going to make your nuclear membrane little bit more permeabilized. Now what does it mean? It means that it is going to loosen the nuclear membrane permeability so that whatever treatment you are giving, it will go inside. Okay, so this will allow the antibodies, for example, in this case, we are using anti-lamin antibody. So it will allow the antibodies, which is used to detect the lamin B proteins, it will penetrate the nuclei of the cells. And that's why you need mild detergent in order to loosen the permeability of the nuclear membrane. So I hope this was clear. It's very simple. Okay, moving on to the next question. Third, describe the effect of CDC2 kinase and protein kinase C on the nuclei. So here you need to talk about all the images what is given over here. So let's see that. So incubation of the cell nuclei with the CDC2 kinase, it results in the disappearance of the nucleus membrane. Okay, if you see that, it gets disappearance of the lamin proteins. Since here the stain what we have used is the anti-lamin B antibody, right? And that's why you see these images. Now here there is no images. So what does it mean? It means what? It means that here there is no lamin protein. So there is over here, it is the disappearance of the lamin proteins as when I compare to the control. So 0R means it is control, right? So here you can see the lamin proteins are present, whereas in the case of figure C, there is nothing is present. Okay. Now fluorescent signals appear around the inner nuclear membrane that are present in the control nuclei, whereas it is absent in the case of C. Okay, because that signal is completely vanished in the case of the C. Okay, now the morphology of nuclei on the phase contrast microscopy. So this is your phase contrast microscopy. This is your confocal. Okay, that is based on the immunofluorescence. So if you see this over here, this is based on the phase contrast. This is based on the immunofluorescence. Okay, so if you see the morphology of the nuclei on the phase contrast micrographs, it did not change as a result of CDC2 kinase treatment. So if you see over here, the nuclear membrane is intact. Now, PKC treatment did not cause any. So here also you can see that the PKC treatment, it did not cause any changes in the appearance of the location of the lamin proteins which are present as compared to the control. Okay. So here also if you see, there is no change in the inner nuclear membrane. So I hope this is clear. So this is your immunofluorescence based micrographs and these are based on the phase contrast microscopy. Okay, so last question. Considering the function of CDC2, what is the physiological significance of the phenomenon revealed in this experiment? This is very obvious even if you have not done the experiment and have listened to my lectures carefully, then also you can answer this. So phosphorylation of the lamin proteins by CDC2 kinase or CDK1, it induces the depolymerization of the nuclear laminar layer. So what will happen? So once your lamin proteins A and C gets phosphorylated, what happens? The nuclear membrane or the nuclear envelope starts disappearing or the breakdown happens during the prophase of the mitosis. So even if you did not do this experiment and would have listened to the lecture, then also you can answer this. So this is how you interpret the data. This is how you read the images which is given. Okay, so I hope this was really helpful to you guys. And still, if any doubts are there, or if you have any data which you are not able to do the analysis, you need certain guidance, please contact me at excelsioracademy1228@gmail.com. You can also message me in the Telegram, which is by named Excelsior Academy itself. And yes, if not none of the options you have, then you can comment in the YouTube channel itself. So I hope you like this video. Thank you. With this, I would like to announce that I have completed the cell cycle with respect to the Cooper, everything depending on the interview type questions, data interpretation, the theory, everything has been done. So maybe in future, if I get the time, I'm gonna take the MCQ also. So till then, take care and keep studying. Thank you.